This is Chapter 2, Part B, Chemistry Comes Alive. So last section we looked at just basic chemistry, so atoms, elements, different types of chemical bonds. This section we're going to focus on biochemistry, or the study of the chemical composition and reactions of living matter, so sometimes called just organic chemistry. So all chemicals are either organic or inorganic. So things that are organic come from living things. Things that are inorganic can just come from the environment, non-living substances. So things like rocks, water, minerals are all inorganic. So these are naturally occurring. Whereas organic molecules can only be synthesized and found in living systems. So water is the most important, most abundant inorganic compound. It accounts for roughly 70% of our total body. So majority of that comes from just the fluid in the cells that keep our cells filled. But it's the most important inorganic compound because it has special chemical properties. So things like a high heat capacity, high heat of vaporization, polar properties, reactivity, and cushioning. So high heat capacity, or sometimes high specific heat, is the ability to absorb heat without a drastic change in temperature. Water is able to absorb a lot of heat before it actually changes temperature. So this is why it takes forever, seemingly, for water to boil on the stove. So it takes a while for the water to absorb that heat and change its temperature. High heat of vaporization also means that the water can hold a large amount of heat before it changes states and evaporates. So this is useful for our cooling thermoregulatory mechanism. So the sweat is only going to cool you off if it evaporates from your skin. So if you wipe off all the sweat, it's not going to do any good to cool you off. So the sweat will absorb the excess heat from your skin, and as it evaporates, it will carry that heat away with it. Water also has polar solvent properties, like we discussed in the last chapter, that water is a polar molecule, so meaning it has a slightly negative end and a slightly positive end. So this allows it to dissolve ionic substances like salt. So remember, ionic compounds are also charged molecules. So these opposite charges will attract. So the negative ends of a water molecule will attract the cations, right, in the salt, and the positively charged ends of the water molecule will attract the anions of the salt. Water has a high reactivity, which makes it a necessary part of our cells' chemical reactions, so hydrolysis and dehydration synthesis reactions. So hydrolysis just means water splitting. Lysis means to split. So we're using water to split molecules. Dehydration means we're removing water to synthesize or make new bonds. Right? So dehydration synthesis, we remove an H2O and we form a new chemical bond to link these two molecules. Right? We could go the opposite direction if we want to break apart these molecules and add water. So the water will split the bond, right, and separate the two molecules. Water also provides a cushioning function, so it helps protect certain organs from physical trauma. So cerebrospinal fluid cushions the nervous system organs like the brain and the spinal cord. Serous fluid helps protect the organs and the other body cavities. So we said salts were ionic compounds, meaning that they carry charges. So they're going to be able to dissociate or dissolve in water. So they separate into their cations, positively charged molecules, and the anions, the negatively charged atoms. So in this example, sodium is our cation and chloride is our anion. All ions are called electrolytes because they carry an electrical charge, which means they're capable of conducting electrical currents in solutions. So these ions play specialized roles in various body functions. So some important electrolytes in the body are calcium, which assists with muscle contraction, magnesium, phosphorus, potassium, sodium, and chloride. So this balance of the ions and electrolytes is very important for homeostasis. Acids and bases are both also considered electrolytes because they ionize and dissociate in water. So they break apart and release charged particles. So acids are considered proton donors, meaning they release hydrogen ions. So hydrogen only has one proton. So when we donate one proton, we essentially donate a hydrogen. So when acids are dissolved in water, so this example, hydrochloric acid, so we split it and we get negatively charged chlorine and a positively charged hydrogen. Right? So we have released a proton to the solution. Bases, on the other hand, are proton acceptors, so they're going to pick up any excess hydrogen ions in the solution. So when we dissolve a base in a solution, it releases a negatively charged hydroxyl ion, OH. pH is the relative acid-base concentration of a solution. The pH scale measures the concentration of hydrogen ions within that solution. So the more hydrogen ions the solution has, the more acidic the solution will be. So here showing our concentration of hydrogen. Right? So here would be smaller numbers, 10 to the negative 14, right, opposed to 1. pH ranges from 0 to 14, with 0 to 7 being on the acidic side, 7 being neutral, and 7 to 14 being on the basic end of the spectrum. Acidic solutions have high concentration of hydrogen, but they have a low pH. A lot of students get it mixed up because you would kind of intuitively think that the more acidic something is, the higher the pH would be, but it's the opposite. So value goes down as acidity goes up. So it's an inverse relationship. Right? Alkaline solutions would have a low concentration of hydrogen and a higher pH value. So this is showing a low pH. So we have an acidic solution. So we have lots of hydrogen in this solution compared to hydroxyl. In a basic solution, we have more hydroxyl ions than we have hydrogen. Neutralization of acids and bases is when they're mixed together and displacement reactions occur. So we form neutral products like water and salt. So in this example, we have hydrochloric acid and sodium hydroxide. So when we mix these together, we're going to split the bonds between the hydrogen and the chloride and split the bonds between the sodium and the hydroxide. So we have our rearrangement, our displacement reaction, and we end up with sodium chloride and water. So relatively neutral, harmless compounds now. Buffers are molecules that resist any large-scale swings in the pH. So we need to maintain those homeostatic levels of pH. So buffers help us to do that. So buffers work by either releasing hydrogen ions if the pH starts to rise, or binding hydrogen ions if it starts to fall. So at a low pH, so it's becoming more acidic, we have too much hydrogen in the solution. The excess hydrogen is going to be bonded to our buffer here. Right? So we're going to take this hydrogen out of the solution and prevent the solution from becoming more acidic. As the solution becomes more basic, so we start to get a higher pH, we don't have enough hydrogen in the solution. Now the buffer will release the hydrogen that it was holding to increase the hydrogen concentration. All organic molecules contain carbon. Exception would be CO2 and CO, carbon monoxide gas, which are considered inorganic since they're found in the atmosphere. But carbon is useful for organic chemistry because it's electroneutral. So it always forms those covalent bonds where it shares electrons. It will never form an ionic bond where it would gain or lose an electron. It's capable of forming 4 covalent bonds with other elements. So this gives it a wide diversity of shape and structure in the type of molecules it can form. So this is unique to our living systems. So the major organic compounds are carbohydrates, lipids, proteins, and nucleic acids. Nucleic acids are composed of nucleotides, carbohydrates composed of monosaccharides, lipids composed of fatty acids, and proteins composed of amino acids. So most organic molecules are polymers, poly meaning many. So a monomer are the individual building blocks of the organic molecules. A polymer would be chains of multiple monomers linked together. These polymers are synthesized by those dehydration synthesis reactions, right? So removing water to synthesize and make something new. They're also going to be broken down by reversing the reaction by hydrolysis. So instead of removing water, we add water to split the bond. So you can think of it in terms of like Legos. Right? So an individual Lego block are your monomers, and then you can put the individual Lego blocks together in almost infinite different formations to make different structures. Right? So say we make a house with the Legos. Right? So then we break down the house, we separate all the building blocks again, and we can use those same building blocks now to make a tower. Right? We can make something else with those same monomers. So organic molecules work kind of the same way. So you have some amino acid monomers that we can link together to make different proteins. So in dehydration synthesis, again, we're removing water, right, to form a new covalent bond. Right? Hydrolysis would be the reverse. So we're adding water to split this bond and separate the two monomers. The carbohydrates include sugar and starches. They're composed only of carbon, hydrogen, and oxygen. So there's three classes of carbohydrates. Monosaccharides, mono meaning one. These are just simple single sugars like glucose and fructose. These are the smallest unit of the carbohydrate. So these are your monomers. Disaccharides contain two sugars, right? So di meaning two, and then polysaccharides contain many sugars linked together. In animals, humans, our polysaccharide carbohydrate storage form is glycogen. We store in the muscles. Plants can use starch, so they're polysaccharides compose starch. So when you get like starchy vegetables. Lipids contain carbon, hydrogen, oxygen, but in fewer quantities than the carbohydrates, right? And sometimes a little bit of phosphorus. But they are insoluble in water. Right? So oil and water do not mix. Three main types of lipids include triglycerides, phospholipids, and steroids. Triglycerides, or neutral fats, are called fats when they're solid and oils when they're liquid. So they're still triglycerides, but they can exist in different states of matter. But they're all composed of three fatty acids bonded to a glycerol. So we get three fatty acids, glyceride for the glycerol. So the main functions of these triglycerides are energy storage, insulation, and protection. So fats have more than double the amount of calories per gram and energy than carbohydrates and proteins. So triglycerides can be constructed of saturated fatty acids or unsaturated fatty acids. So when something is saturated, it's soaked, right? It's full. So saturated fatty acids are soaked full with hydrogens. They have the maximum number of hydrogen atoms allowable by that molecule. So these saturated fatty acids would be solid at room temperature. So example would be like butter, animal fat. Unsaturated fatty acids have one or more double bonds in the fatty acid chain in the tail here. So that's going to put little kinks in the tails of the fatty acids. So we're not able to pack them in as tightly. They're more kind of loosely arranged. So these would be liquids at room temperature. So trans fats have been, or oils that have been modified. So these are the unhealthy fats. So things like margarine, shortening. So they take vegetable oil and kind of synthetically add hydrogen to it to make it more solid. Omega-3 fatty acids, like found in fish oils, are the heart healthy, the good types of fats. Phospholipids are just modified triglycerides. So instead of having three fatty acid tails, it only has two. And we've added a phosphate group. So we get a phospholipid, so a phosphate lipid. So there's two different regions of the phospholipid. You have a head region and a tail region with the fatty acid chains. The head is considered the polar region or the hydrophilic. So this is the water-loving end. In the cell membrane, right, we have this double layer structure of phospholipids. So The heads are going to be exposed to the fluid outside the cell and the fluid inside the cell. The tails, however, are hydrophobic, so they are scared of water. They don't want to be exposed to water, so they will kind of naturally turn in on themselves and form this barrier and this membrane. Steroids consist of 4 interlocking ring structures. So the most common steroid is cholesterol, and from that we derive all the other steroid hormones. So it's building block for vitamin D, steroid synthesis, and bile salt synthesis. It's also important for the cell membrane structure. So some of the steroid hormones, stero is generally going to be kind of your root word. So if you see stero in there, then we're talking about a steroid, right? So aldosterone, testosterone, progesterone, but they're all based on the standard 4-ring structure. Steroids and cholesterol are also important for the myelin sheath and nerve cell homeostasis, nerve cell transmission. Proteins have by far the most varied function of any of the biological molecules. So there could be structural proteins, there could be chemical proteins like enzymes, contraction proteins, motor proteins like in muscles. So these polymers of amino acids, these proteins, are held together by what's called a peptide bond. So it's still a dehydration synthesis, so we're removing an H2O to form a new bond, but now it's just referred to as a peptide bond to show that we're synthesizing proteins. All proteins in the body are only made up from about 20 different types of amino acids. So these amino acids, like we said, are joined by covalent bonds called peptide bonds. So one amino acid has an amine group and an acid group. So this is why they're called amino acids. The only component that makes them different is this side chain, this R group. So these are the different possible side groups that could be attached to designate the different amino acids, but they all have this same kind of basic structure. 4 levels of protein structure determine the shape and the function. So again, remember, everything in anatomy and physiology, the function is determined by the shape. So this is also true on just the molecular shape of these proteins. They're going to dictate what functions they can carry out. So the first level of protein structure is primary. So this is just a sequence or chain of amino acids linked end to end. So once the chain starts to get a little bit larger, it will progress to the secondary structure, which is either a beta sheet, so it just kind of wraps around, or an alpha helix, where it will coil up into this helix shape. Tertiary protein structure occurs when we have alpha helices or beta sheets kind of combining together now to make a larger structure. And then quaternary protein structure is when we have more than one of these tertiary structures. We have many helices and sheets together, forming a globular protein. So there's 2 general shapes of proteins, fibrous or globular. Fibrous proteins are generally your structural proteins, things like collagen and keratin. So they're strand-like in structure, which makes them more stable. So they have good capacity for mechanical support and tensile strength. Globular proteins are more compact and literally just kind of like little globs of these amino acids and these proteins. So these would have the tertiary or the quaternary structures. So they also have specific functional regions, things like binding sites. So hemoglobin can bind oxygen. Some enzymes that are proteins bind specific substrates. Antibodies bind specific antigens. Hormones bind to specific cells. Protein denaturation occurs when those proteins unfold and lose that functional 3D shape. So as a result, their active or binding sites are going to become deactivated because we change the shape of the lock, so now their normal key will no longer fit in it. So this could be caused by a decrease in the pH or increased temperature. So generally, if the changes are extreme, then it would be an irreversible reaction. So example would be frying an egg, right? So albumin is the protein in egg white. When you cook an egg, you're increasing the temperature and denaturing those proteins and unwinding their structure. So then they're going to form new chemical bonds and cross-link to transition to that solid egg white appearance. Right? And irreversible is in, once you fry an egg, you can't go back and unfry it. Enzymes are special globular proteins that act as those biological catalysts to help speed up the rate of chemical reactions. So they do this by lowering the activation energy needed to initiate the reaction. So, example, without an enzyme, right, we would have to push the ball up this steep hill to get it to go down to our product. With an enzyme, it makes that hill just a little less steep, so we don't have to use as much energy, right, to get our ball rolling. Enzymes are specific, so they're going to act on specific substrates, meaning kind of like a lock and key mechanism. So only the correct substrate will fit in our binding sites. Enzyme names usually end in "ase", and sometimes are named for the type of reaction they catalyze. So like lipases break down fats, lactases break apart lactose, protease breaks down proteins. DNA polymerase is an enzyme that's going to make DNA polymers. Amylase is an enzyme in saliva that starts to break down starch. So when you eat a piece of bread or for a minute without swallowing it, it will start to taste sweet because the amylase is breaking down the starch into single glucose molecules. Nucleic acids are the largest molecules in the body. So DNA, if we were to unwind it from end to end out of the cell, it would be about 6 feet long. Nucleic acids are polymers made up of monomers called nucleotides. So nucleotide is composed of a 5-carbon sugar, a phosphate group, and one of 4 nitrogenous bases. So the bases are like the A, T, C, G in DNA, those 4 letters. 2 major classes of nucleic acids include deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. DNA is what holds our genetic blueprint for all proteins. So basically the instructions on how to make one of you is in your DNA. Right? So DNA is a double-stranded helix molecule, right? So it's kind of a twisted ladder structure. The nucleotides contain the deoxyribose sugar, right, our phosphate group, and one of those 4 bases, A, T, C, or G. So the backbone of the DNA molecule is just repeating units of sugar, phosphate, sugar, phosphate, sugar, phosphate. The rungs of the ladder are composed of the bases when they pair up together. And remember, we said these are held together by those weak attractive forces in the hydrogen bonds. Right? So there's no actual electrons interacting between the 2 strands. There are 2 groups of nitrogenous bases in DNA. So we have purines and pyrimidines. Purines are double-ringed molecules, and the pyrimidines are single-ring structured. So in the DNA molecule, we'll always have 1 purine bonded to 1 pyrimidine. Right, so A always binds with T, C always binds with G. You will never have A bonded to G or T bonded to C unless there's a mutation. So DNA follows this complementary base pairing rule. So again, A always pairs with T, C always binds with G. So if they were to try to pair up with the opposite partner, they wouldn't fit together. So the genetic code, this blueprint in your DNA, is held in the sequence of the bases or the sequence of the letters in the DNA. So TTCA codes for a different protein, would be a different gene than GGCT. So the genetic code is the sequence of the letters. RNA is kind of the intermediate between DNA and protein. So it links DNA to protein synthesis. Even though it's a nucleic acid, it does differ slightly from DNA. So one difference is that it is single-stranded. DNA is double-stranded because we need a replication mechanism to make copies of DNA. RNA is just a temporary copy, so we make a temporary copy of a gene to make that protein, and then we're done with the RNA. So we don't need a double strand to replicate the RNA like we do with DNA. Another difference is instead of thymine pairing with adenine, it uses uracil. So it functions in the place of thymine in RNA. There are also 3 types of RNA that work together to carry out this process of protein synthesis. So there's messenger RNA, which is going to basically carry the message encoded from the DNA. Ribosomal RNA, which is going to translate this message, and transfer RNA, which is going to transfer and deliver amino acids to the growing protein chain. Chemical energy released when glucose is broken down is captured in ATP, or adenosine triphosphate. So this is the primary energy molecule of the cell. So technically it is considered a nucleic acid because it is an adenine nucleotide with 2 extra phosphates attached. So if we were to cut it off here, this would be the part of DNA. So we have, or RNA. So we have our sugar, phosphate, and a base. So we add 2 more phosphates on there to carry more energy in the molecule. So this offers immediate usable energy to the body cells as needed. So when we snip off this 3rd phosphate, it's going to liberate the energy that was holding these atoms together. So again, it's the terminal or the 3rd phosphate group of ATP that transfers the energy. So a loss of the phosphate group would convert the ATP to a DP, diphosphate. So we have 2 phosphates. Loss of a 2nd phosphate, then we would just have our adenine nucleotide, or AMP. So you can think about it kind of in terms of a battery. So we can store energy in the battery when we charge it by adding that 3rd phosphate group. So then when we need some energy, we use the battery, we cut off that 3rd phosphate group, we use the energy, but now we have a partially charged battery. So 3 examples of different types of cellular work that can be done by ATP. So first is transport work. So ATP can help to pump solutes and ions across the membrane, kind of going upstream against the natural current or flow. Mechanical work. ATP causes muscle cells to contract. And also chemical work. So ATP can provide energy to drive our biochemical reactions.